biggest limitations and challenges that comparative oncology faces, one, have to do with numbers. And so a lot of times I'm at Texas A&M and I am an island unto myself, and I can take in the number of lymphomas that come in my clinic, but that's about it. And so I may not be able to accrue numbers to look at these cases in a reasonable period of time. In the human side, there are huge cooperatives that do these trials together as multi-institutional facilities that can get this accrual in a very rapid period of time. We have one in veterinary oncology, and so the Comparative Oncology Trials Consortium, which is run through the National Cancer Institute, has, I think, over 20 universities or practices that are associated with it, and we are able to run trials through there. And in fact, we've run quite a few, and we at A&M have participated in quite a few of them. And so that is sort of a start, but we need to expand that. It needs to be easier. The infrastructure needs to be there in order for this to be routine. And I think earlier on, the drug companies or collaborators that want to run these trials need to see the benefit of doing it bigger earlier. I think a lot of times they come and they've got X amount of money and they want to do these small studies, which are great for proof of concept, but that's it. And so if we can convince them to take that bigger leap a little sooner in the process, using the multi-institutional approach, I think that will help. Other things are the types of diseases. So for example, melanoma. It's common in dogs, it's common in people, right? It's the #1 growing cancer in women over 35. Really common, but they're not the same disease. So dogs, when they get melanoma, it's like mucosal melanoma, which represent a very small fraction of the human melanomas, which tend to be UV induced. And so knowing the model and knowing what's appropriate. And so on the human side, these UV induced melanomas often have a BRAF mutation, which responds to BRAF drugs. But our canine melanomas don't. They don't tend to have that mutation because they're not the same disease. However, bladder cancer in dogs often does have this BRAF mutation, the exact same one. And so maybe knowing molecularly and more about the cancers, which we are just starting to learn, to find out what's the appropriate model for the disease. So maybe canine melanoma isn't correct to study human melanoma. Maybe canine bladder tumor is the better model to study human melanoma. Same thing goes with our c-KIT mutations, and so we see that in mast cell tumors all the time. Humans almost never get mast cell tumors, but they do get GISTs or gastrointestinal stromal tumors, which also tend to have c-KIT overexpression and mutations. And so maybe canine or feline mast cell tumor is the right disease to study for human GIST. And so just learning more about the molecular makeup of our tumors, what's driving them, thinking about them less in an anatomic sense and more in a genetic sense, which pathways are altered and those kinds of things, I think will help us drive it forward. But right now we're in the very early stages of really fully understanding the molecular mechanisms behind what drives our cancer in our veterinary species.